I'm going to spend the next hour or so talking about a couple of issues that are pretty important with the whole CRISPR technology. The first one is, well, I know what I was going to say. I don't actually live here. I live in Utah. I'm in the biochemistry department at the University of Utah. And so why am I doing this? Well, I was here on sabbatical last year, and I was part of the group that hatched the idea of having a CRISPR workshop. And when they were asking for people who would like to organize the workshop, everybody else stepped back. And, and then when it came time to do the workshop again, there were no volunteers. We finally twisted Martin's arm to come and help with it, particularly on the informatics side. But just in case you mistake who I am and what I'm doing here, I'm an interloper, but I was here last year on sabbatical. Okay. The two issues I'm going to talk about are delivery of the nucleases and off-target effects. So, delivery. Every organism, every cell type is going to have its own requirements for how it is going to accept the nuclease that you want to put in. And all of this nuclease technology piggybacks on vectorology and transfections and transformations that people have worked on for years and years. But there are some advantages to doing things in different ways, and I'm going to tell you about the way we're going to do it this afternoon or during this week. So just to start out, there are really good vectors available for delivering Cas9 and guide RNA, and I'm going to illustrate a couple from Feng Zhang's lab. They've developed that one's not working at the moment. They've developed a lentiviral vector that has a Cas9 coding sequence and a segment in the vector that has the body of the guide RNA, the downstream portion, and then a stuffer that can be replaced with a short oligonucleotide pair to introduce any particular guide sequence, any 20 nucleotide or approximately 20 nucleotide guide sequence. And there's an RNA polymerase III promoter that'll drive production of the guide RNA, and a strong mammalian cell promoter. This is an elongation factor 1 alpha to drive production of Cas9. And they also have plasmid versions, the Cas9 coding sequence. This particular one has a very small stuffer, and I don't know how many of you are familiar with the Golden Gate technology, but there are these restriction enzymes that are actually like FOC1. They cut some distance from their recognition sequence and create an overlap. And the overlap is in an arbitrary sequence. So you can have a situation like this where you've got two sites for this enzyme BBS1, but it'll cut within the vector sequence that you want to retain. Then you just make an oligonucleotide pair that has corresponding overlaps to the ones that are generated by BBS1 on the two sides, and your guide sequence in the middle, and just drop it in with a simple ligation reaction. And there are lots of variations on this. So people have used vectors of various kinds, expression constructs. You can, in some systems, it's actually more convenient to deliver the Cas9 as a messenger RNA rather than as a DNA. But how you do this is going to be constrained to some extent anyway by the cells or the organisms that you're delivering to. And an approach that has proved effective in a lot of different situations, although probably isn't useful everywhere. I know of one situation where it hasn't worked yet that's causing me headaches, is to make the Cas9 ribonucleoprotein complex in a test tube and then deliver the RNP to cells. So what you're going to do today is to produce a DNA template for in vitro transcription of the guide RNA by using T7 RNA polymerase. We'll provide Cas9 protein, and you can make this RNP complex. Today, we're not using a donor DNA, but you can incorporate that, and then just introduce this into cells using electroporation or some other sort of transfection method, or in organisms where things are being done at the level of the embryo, you can physically inject the RNP into mouse embryos, fish embryos, frog embryos, as a method of delivery. And although today we're going to transcribe this RNA ourselves, you can also buy a guide RNA from a company. They can chemically synthesize the guide RNA. The guide RNAs we typically use are 100 nucleotides long, approximately, and that's accessible by chemical synthesis. If you do chemical synthesis, you can put some variations into the RNA. You can protect the ends from degradation by incorporating modified phosphodiester linkages. You can put in non-natural bases if you want to. You have a lot of control over what the RNA looks like if you order it from somebody by chemical synthesis. So one of the reasons for using RNP is that it's really a hit and run type of delivery. And this is from a paper from Jinsoo Kim's lab. So if you deliver Cas9 and the guide RNA on a plasmid or two plasmids, it takes a while to produce the nuclease. And it's quite a number of hours after you started the experiment before you begin to see any mutations. These are T7E1 assays up here. So even by 3 hours, you don't see anything. A little bit at 6, and then it goes on, and you keep producing mutations over a period of time. This is just looking at indels. With the RNP, electroporating it into these same cells, you can see a little bit of mutagenesis within the first hour, and things are finished within about 12 hours. In addition, the RNP is gone. The protein is gone within a day. The plasmid-introduced Cas9 continues to be present for a while. The DNA is there, the message is there, the protein is there, and all of them have to be eliminated somehow before you really get rid of the protein. And it's this tail that may be creating some of the off-target effects that we try to avoid. Okay, so this is one reason for using the RNP. Yes?
So this might become critical when you're actually trying to send a donor template in, because if your enzyme is producing and it's not produced till 3 hours, but you're giving the donor DNA simultaneously, does it affect the way it works? So is it better to use RNPs when you do big donor templates?
It could be. I don't know that anybody's looked at that particularly, just whether there's an issue about timing. If the donor is there for a long time before the cuts are actually made, is this going to reduce the effectiveness of using the donor? And I don't know the answer to that. With any of these things, if you're working in cultured cells, you can stagger delivery if you want to.
Just kind of more general thing that would follow up to that we talked about is whether the donor is limiting.
Yeah.
Do you have insights on that? Like, have you titrated?
I haven't particularly. Dan Voitas has a nice story about that, where I believe he found that in the plants he was working on, that the donor was in fact limiting. But you should ask him, because there's a 50-50 chance I got it backwards. But you can separately titrate the amount of donor and the amount of nuclease.
Because usually they say that you have to if you are delivering it as a part of a plasmid backbone, it's better if you linearize it.
Mm-hmm.
But I'm not sure how much is the stability of these linear fragments inside the cell while these is being produced. So.
And we were talking about that earlier with Lynn. One of the disadvantages to using a linear donor is that those ends have some tendency to integrate randomly in the genome. But I go back to a paper that was published by my friend John Wilson in about 1984. He pointed out that with many of the methods you use to deliver DNA to cells, what you start out with is not what ultimately gets into the cell. You know, these DNAs have to traverse the cytoplasm, get into the nucleus, and there's both cleavage and joining that's going on. So you send in a linear, some of those ends are going to be joined You got a circle again. You send in a circle and there's going to be some breakage.
So what about the NLS in these target donors, like nuclear localization?
You can't put a protein NLS on a DNA. So
Not like a sequence, like a nucleotide sequence? Does it need it?
so DNAs don't need a special sequence to get into the nucleus.
Okay. But the time might be important.
Timing might be important. Okay, I'm just going to give you one example to sort of validate RNP delivery. And this is an experiment that Mark DeWitt and I did while I was here. Yeah?
I have a question about the linear donor. Like your question, they integrate to the genome. But I think for each linear donor, we should add like a TK outside the reading frame.
Right. So,
Do the double selection.
right. The nice thing about nuclease-mediated homologous recombination events is that they're so efficient. I mean, even if you're saying, oh gosh, it's only 20%, 20% of the cells is hugely efficient, and it's more efficient than random integration. So typically we don't design our vectors the way the old Kopecky method did. We don't put in a positive and a negative selectable marker because we don't need them. All right. So what Mark and I did was we established a BFP gene in the genome of these HEK293 T cells and generated a donor that would convert the one codon that distinguishes GFP from BFP, and then delivered Cas9 with a guide RNA that cuts essentially right on top of that codon. And then faxed the cells. And we delivered the RNP. We delivered the RNP by nucleofection, as you will be doing tomorrow afternoon. And the starting population should have only blue fluorescence. If all we do is knock out BFP with non-homologous end joining, they should have no fluorescence. But if we convert them to GFP by utilization of the donor, we should now have green fluorescence. And the cells we started with were 94% BFP positive. There are a few laggards that didn't have any fluorescence. But after this RNP delivery, there's 1% of the cells still had BFP fluorescence. 74% were knockouts, and 25% now had GFP fluorescence. So this is what we refer to as a typical experiment, which means it's the best one we ever did, showing that RNP delivery, at least in these 293 T cells, was very effective. Okay, I'm going to spend a lot of time talking about off-target effects. So the first thing, well, I guess did I have the slide I'm thinking of is somewhere else in the list. Okay. First thing we're going to talk about is how bad are off-target effects with the CRISPR tools. So there were several papers published in late 2013 that essentially said, oh my God, this is terrible. You know, every place in the genome is getting cut, you know, can't use the CRISPR system. It wasn't that bad, but there was a lot of attention stirred up about the lack of specificity. And so this is a paper from Keith Jung's lab, and what they're doing is they've got cells that have a single copy of GFP in the genome, and they're attacking it with different guide RNAs. These are 3 different guide RNAs, and just looking for the production of GFP negative cells. And the efficiency of making GFP negative cells relative to a perfectly matched guide RNA, perfectly matched to the target, is being measured here with single base mutations in the guide RNA relative to the target, or 2 nucleotide alterations of various kinds. So here's a target, a guide RNA hitting a particular target, where if you make a single base alteration in the guide RNA, almost anywhere within that guide RNA, you're still getting about the same level of mutagenesis. You get right up next to the PAM, and it seems to care a little bit. Okay? This guide RNA is much more sensitive, particularly to sequence mismatches that are close to the PAM, but out here, not so sensitive, and this one is kind of intermediate. So this is the kind of evidence that people were getting that said, whoa, wait a minute, maybe we can't make distinctions.
Is this on a plasmid or is it integrated?
This is integrated in the genome. Single copy. They then went and looked at a particular target. Actually, I think there were several targets in this paper. In the genome, this is the target, that's the sequence that the guide RNA is going after. And then they searched the genome for related sequences that might also be cut using this same guide RNA. And in several different cell types, they're getting pretty good levels of mutagenesis. This is just end-join mutagenesis. These are indels. But you look at some of these secondary targets, and there are ones that are mutated as frequently as the design target, and one down here that's even mutated more often. So again, this was sort of raising a red flag and saying, you know, we have a specificity problem. And although it's not identical across the different cell types, it's still an issue. This paper is from Feng Zhang's lab, and they found something similar. They took a particular target and ran mutations through the guide RNA, sequence changes through the guide RNA, and just looked to see whether those guide RNAs would still mutate this chromosomal target in the EMX1 gene. And without explaining everything that's going on here, the design target is indicated by the sequence with these dark gray boxes. If there's a white box, that means if this G was substituted with a C, then there was no cleavage, no mutagenesis. But if you're getting a dark blue box, that means you're getting a very high level of mutagenesis, even with the guide RNA that has that nucleotide at that position in place of the canonical one. Okay? So they again are seeing, particularly as you get, the PAM is over here, particularly as you get away from the PAM, there's quite a bit of mismatch tolerance. And this is looking at like the Zhang lab did look at some natural sequences related to the design sequence and finding at a few of them that you're getting indel frequencies that are a little bit worrying. One of the things they did here was to look at the influence of nuclease concentration, or at least delivery, the amount being delivered, on the specificity. So of course, as you increase the amount of Cas9 plus guide RNA plasmid, in this case, you're getting more mutagenesis at the target, but you're also getting more at secondary sites. And what they're plotting here, and we'll see some other plots like this, is specificity, which is a ratio of on-target to off-target mutagenesis. Okay? So if you go to low levels, you have more specificity, but you have a lower level of absolute mutagenesis at your design target. So this is another indication that if you have the nuclease around for a longer time, you're going to get into deeper trouble. Okay? More nuclease, more opportunity to hit the secondary sites. Now, it seemed clear to me that mismatch tolerance was adaptive for natural CRISPR systems, because when a virus infects the cell, since viral genomes evolve at a high rate, the genome that comes in may not be absolutely identical to the one that set up spacers in this CRISPR array. It may be a close relative, but not identical. So in order for the CRISPR system to work effectively, a given spacer has to be able to attack a sequence that's identical to the original agent, but also its close relatives. So the tolerance for mismatches may be adaptive, and just using the natural CRISPR components, maybe you can't get around it.
What do we know about the Cas9 expression when you have invasion? How high is it?
In the natural CRISPR system, I don't know the answer to that. Have anybody here who's a CRISPR maven? Yeah, I don't know what the level of any of those components is in a bacterial or archaeal cell naturally. Okay, so before going on, I think we need to, this is the slide I was looking for earlier, need to think about how much we care about off-target cleavage. Well, it depends on what you're doing, obviously. So, whoa. If you're working in a model organism and you're mutating a gene or altering a gene so you can study aspects of its function, you want to make sure that the mutations you made in that gene, the sequence changes you made in that gene, are what's responsible for the phenotype that you're ultimately examining. And you can do this by making independent mutations in the target, which are unlikely to have identical off-target mutations. And you can also clean up the background in a genetic organism by backcrossing. So you have workarounds. You care, but you have workarounds. If you're working in food organisms, like some of my friends are, you're concerned about safety, but you have the opportunity to do whole genome sequencing when you can go through a cloned stage, which you typically can for livestock and crop plants. And you can do very careful phenotyping. So you can look to see whether there are other things going on, but then it's going to be necessary to make sure that you haven't messed things up just by making sure that the organisms are continuing to do what you expected them to do. When we talk about clinical applications, the clinical applications that are being pursued currently are somatic therapies, where you typically are taking cells from a patient and putting them back into a patient, or generating a panel of cells that can go into patients. And so you're working with people who have limited lifespan after the therapy. So what do you care about? Well, it'd be a bad idea to turn these therapeutic cells into aggressive cancer cells. But other than that, what do you care? If you knock out if you're doing a therapy for a blood cell related disease and you happen to knock out a specific, muscle specific collagen of some kind, do you really care? So there are places where you can be tolerant of things that are happening off target. With stem cell therapy, I mean, these are things that people are working on. I would say currently there needs to be a lot more work on the cells themselves if they're going to be used for therapy, and we'll deal with those situations. Again, those would be somatic therapies. But for germline therapy, which people have begun talking about for humans, then it's a much more serious worry, because any modification that's made in the germline of one individual will necessarily be passed on to that individual's descendants. And I hope Jonathan Weissman on Friday will talk about some of these issues. Okay, so there are a bunch of ways that people have used to try to see what's going on at secondary targets in the genome. One is just to use sequence homology or some sort of experimental determination of the specificity of your nuclease, and CLEX is the name that people give to this procedure, to predict what the sites are going to be. But there are also experimental methods to go capture those sites. These two methods, and I'll describe GuideSeq more thoroughly, these two methods depend on capturing a linear piece of DNA at a break by non-homologous end joining. Okay? So you capture a piece of DNA and you can go find out where it went in the genome. This is a complicated procedure. I'll tell you a little bit about it, and I'll talk about these two as well, but not about that one. But I'm giving you references just so you can see sort of what's out there. And there's a paper from Jin-Soo Kim and colleagues in this journal called Molecules in Cells that makes at least an analytical comparison of these different methods, and you can go look at that if you want. Okay. So GuideSeq. Keith Jung cannot produce a method without giving it an acronym, and it has to be an easy acronym that people will remember. So he made open with the zinc fingers, and he made, what was it called? code or something, and he's made also true guide RNAs. He has to have an acronym. And you have to go to the paper to see what GUIDE stands for. The way this works is as follows. You introduce into cells a double-stranded oligonucleotide. I think it's on the order of 35 base pairs long. And it has its ends protected. If you don't protect the ends, you don't get it captured very efficiently because it just gets degraded in cells. But with protected ends, it gets end-joined into places where a break is made by whatever nuclease you're using. You can then use the sequence of this little oligo to do amplification. You just amplify all the sequences that are next to that oligo in genomic DNA that you recover after a day or two in the cells. And you can do complex multiple PCRs and get this ready for high throughput sequencing. And what you expect is the sequences that you get from the sequencing to end at the position of any particular break. And because the oligo couldn't go in either orientation, you expect a sequence from that break, out from that break on either side in this high throughput sequencing. And this is the kind of data you get. And let's just look at a particular example. So these are the number of sequence reads from these genomic sequences. And there's the designed sequence at the top. It's got the PAM, NGG, and this is the guide sequence. And so you look at the number of reads for different genomic sequences. In this particular case, the design sequence, the perfect match, came in third. So that wasn't a very specific guide RNA. And so the end position is highly variable, that's expected. But this particular guy that gave the most reads has two mismatches. One way out at the end where we think those mismatches aren't detected very well, but one at an internal position. Even the second one has three mismatches. So GuideSeq told us that, yeah, we do have a specificity problem because in an unbiased method of picking up where the breaks are, we found all of these other sequences. There's a peculiarity about the GuideSeq data. This is looking at GuideSeq reads and where they end in genomic sequences. And you can't really see this, but each little white dot there is a single base. So the GuideSeq reads are very tightly clustered around a particular nucleotide or two in the genomic sequence. And this is probably because with the end protection on that little oligo, the capture oligo, you're not getting any degradation of that oligo, so it can only do blunt joins. So if the genomic sequence is processed, maybe you can't ligate to the oligo anymore, or if it's processed very far. And just for comparison, the natural junctions, and these are now 20 base pair steps, the natural deletions cover quite a wide range of sizes. So there's something different about exactly what's being captured by GuideSeq. And I keep asking Keith Jung and Shengdao Tsai, who's first author, about this, and I've finally given up because they go to the sites that they see are being mutated, secondary sites, and they evaluate them with one of these T71 type assays or deep sequencing of those particular sites. And what they get out of GuideSeq is pretty much validated. So I've stopped arguing. This breaks labeling enrichment on strepped out and next generation sequencing. Bless. Well, these guys needed an acronym too. So I like this procedure because it doesn't depend on any processing of the breaks in cells. So what you do is you treat cells and fix them and deproteinize the nuclei. And then you take one of these hairpin oligonucleotides and you ligate it to whatever ends are present in the DNA from those cells. You capture you shear the DNA afterwards. You don't want to shear it before because you don't want to pick up random ends any more than you have to. But you shear and then capture these ligated structures using, I think this is using biotin and streptavidin beads. Then you ligate a different hairpin oligo onto the other end, and then you can PCR these up and sequence them, do next generation sequencing. And so this is a way to capture breaks that are present in cells at any particular instant of time. And Feng, and this is again from one of Ayn Rand's papers, Feng and his lab have used this procedure to look to see where they should look for real mutations. So these are places breaks are made. You expect mutations there. And so this is just comparing on and how does this go? I think this is on and off target mutagenesis based on the BLESS data, based on chip data. This is just asking Cas9 with a guide RNA to see where it binds in the genome, which is known to be a bad approach, and then sequence similarity. And there are examples where BLESS does a much better job of identifying the worrisome targets than simple sequence similarity does. Okay, so that's another method. This is this HTGTS. Again, I probably mess it up if I tried to tell you what HTGTS stands for, but it's another high throughput method. What it depends on is not very well illustrated. This figure is from this paper by Frock et al. This is from Fred Alt's lab. What it depends on is when a break is made at the target, if a break is also made somewhere else in the genome, you have the possibility of a translocation, the joining of one end from one break and another end from another break. And so one of these T's, high throughput genomic translocation sequencing or something like that. This T is for translocation. So what it's doing is it's sort of like GuideSeq, except it's using on-target ends to capture the off-target sites. Okay? Because end joining probably is going to prefer to join the two ends from a single break back together, but sometimes it'll make a mistake. Okay? What? You,
I just don't know how would you know where the breaks are going to be if they're already.
You don't. So what you do is you, just like in GuideSeq, you use the design target for a PCR. This is linear amplification PCR, and you just ask by high throughput sequencing what got ligated to the target. Okay, and you can do that from either end of the target, and these little fountains describe the various translocations that they picked up using this procedure. Okay, we've got really inventive people in this field. And this is a scheme that was developed in Jinsoo Kim's lab. They call it digenome-seq. So the fundamental process is this. You isolate genomic DNA from whatever your cells or organism happen to be, and you do whole genome sequencing. And when you do that, you expect to see reads that cover every spot in the genome, go across every single internucleotide pair. But you take that same DNA and you cut it with Cas9. Jinsoo often calls Cas9 with a guide RNA RGen, which is an RNA-guided endonuclease. That's what that's for. So if you see RGen, that's what is meant. You cut that same DNA with Cas9 and a particular guide RNA. Now you expect all your sequence reads at the design target to stop. And you can use as much Cas9 as you want so that you get 100% cleavage of the design target. So you get all the reads through the design target stop at the cleavage site. And that's shown here. Mock transfected cells read across everything. This is DNA from the cells that was cleaved in vitro, and almost all the reads stop at the cleavage site. Okay, so if you do this genome-wide, that should identify places where Cas9 with that guide RNA can cut in addition to the site you wanted cut. So, so this identifies those secondary targets. And you can do, although they don't always do, introducing the guide RNA and Cas9 into cells and then doing this same procedure. And then there should be some sites where you might find the, I know what. After repair, then you can't cut in vitro with the nuclease again, so you'll have reads that cross that site. So that gives you some indication of how much indel formation there was at the site, because you can't cut it when you try to cut it in vitro. Okay? They don't always do that. Sometimes they just use this portion to identify secondary targets and then individually interrogate them by PCR and sequencing. It's also true, so things we've been talking about mostly have had to do with base pair mismatches, but it's also been identified that some bulges, either in the DNA strand or the RNA strand, are tolerated, at least in vitro, by some of the guide RNAs. So there are a variety of types of secondary targets that might cause problems. So the bottom line on this is someone should just take all of these strategies for identifying secondary target sites and compare them on the same samples, but nobody's doing that. So I'll just tell you that both the IDLV capture and the guide-seq are cumulative. So you introduce the break and the little capture oligonucleotide, and then you wait for a while. So it may be that it's going to overemphasize some of the secondary targets because what's going on at the design target is over quickly, but if the nuclease stays around, you'll continue cutting the secondary targets. I should cross this out because I'm not worried about that anymore. This HTGTS, the junctions look normal. That's one nice thing about this, because you're getting natural junctions between the target and the ends from the secondary target. But it depends on having at least 2 breaks per cell. So again, it may be overemphasizing some of these off-target sites. Digenome-seq requires very deep sequencing. BLESS, I like BLESS, as I said, but it's a snapshot. And the point in time where you actually do the experiment may influence the outcome. You know, what breaks are there very early after transfection, what breaks are there later on. So they all have their pluses and minuses, but these are methods that are available to look at off-target sites. Now we're going to talk about ways you can avoid hitting the off-target sites. So one way that came up pretty much right away, Prashant Mali and Anne Rand from the Church and Zhang labs have papers describing paired nicking, where you take advantage of a fact that I told you earlier, that you can independently mutate the 2 active sites in Cas9, and what that does is it makes Cas9 into a single-strand cutting enzyme, a nickase. And so if you make the Cas9 that still cuts the RNA-bound strand by mutating this site in one of the ROV-C domains, and you provide 2 guide RNAs on opposite strands that bind to opposite strands, you can make 2 nicks. And as long as those nicks are close to each other, the cell will process that DNA as if it had a double-strand break in it with pretty good efficiency. And this just shows that, this is from the Mali paper, that this works for end-join mutagenesis and also for homologous repair, although I'm going to tell you a little bit more about that momentarily. And this is from the Ayn Rand Feng Zhang paper. And one of the surprising things about this is that it works even when the 2 nick sites are as much as 100 base pairs apart. And you would think 100 bases of overlap, that should hold together really, really well. Why would that ever be looked at as a double-strand break? And I have at least one idea about that, but I don't know. I frankly don't know the answer. The nice thing about paired nicking is it requires recognition by 2 separate guide RNAs. So if one guide RNA can recognize 20 base pairs in the target with a little bit of slop, and the other one recognizes 20 base pairs in the target with a little bit of slop, it's unlikely that the slop for the 2 would appear at the same place in the genome, so you'd make nicks, paired nicks, elsewhere than the designed target. So this is just showing these are specificity ratios for wild-type Cas9 compared to the paired nicking, and this is a log scale, so paired nicking gives a specificity ratio of 100 to 1,000, and for the double-strand break producing Cas9, the specificity ratio is much lower. So it's a way to gain specificity. Yeah?
Have these been compared between, have mechanisms like this for improved specificity been compared between mitotic versus post-mitotic cells? Because that becomes important for clinical applications as well.
Yeah. So there, yeah.
Okay.
No. So I think it's an important question. You know, to what extent do you depend on cells progressing through the cell cycle for any of these phenomena? And that hasn't been very thoroughly studied. One of the things that I'm quite interested in is the extent to which the chromatin structure of a particular target influences how well that target is cut. And the way we've avoided answering that question in the past is by saying, well, most of the time these cells are dividing, they're going through S phase, chromatin is disrupted throughout the genome, and people can make this work without worrying about that issue. So it's another issue that comes up with respect to non-dividing cells, but people have not studied that very thoroughly.
But
So,
don't they, in some studies, don't they arrest the cells in a particular phase of cell cycle to favor one kind of mechanism over the other?
yeah, so Sangamo early on began arresting cells, I think it was at the G2M boundary, and then releasing them so that they would go through S phase and be somewhat synchronized in the period of time when the nuclease and the donor DNA were there. Almost nobody does that anymore. And the paper from the Doudna lab that showed that worked to some extent with the RNP delivery worked at a couple of sites and didn't work at some other sites, and or cell types. And so it's not something that you can rely on all the time, I would say. This is just showing homologous repair with double nicking as composed to single nicking. Single nicks do stimulate homologous recombination compared to no cutting at all, but it's really lousy compared to double nicks. This shows that double nicks using different combinations of the 2 guide RNAs work pretty well, and a 5' overhang works better than the 3' overhang. And I would say that for me initially, this was backwards. And we can talk about that some other time if you want, but I think I now understand it better. And then this is just comparing a single nick to a frank double-strand break. So double-strand breaks and paired nicks work much better than single nicks. This is something I don't want to spend any time on. Keith Jung's lab took dead Cas9 and linked it to the FOC1 nuclease domain, turning it into sort of a poor man's talon, And I don't think it's kind of complicated, and there's no real reason to do it except that you could get what's effectively paired nicking because you have to dimerize with RNA recognition rather than protein recognition. And this is something from Jinsoo Kim's lab again, and there are a variety of things going on here. One of the things I wanted to point out is that they, for a different reason, they began looking at guide RNAs that had a mismatched G, a matched G, or 2 mismatched Gs at the 5' end of the guide. So the PAM's over here. At the other end, if you have 2 extra Gs, it turns out that the on-target effectiveness for several targets is pretty close to the standard guide where all 20 bases are matched. But there seems to be less activity at the off-target sites. And so just comparing the blue and the red at the on-target and off-target sites for several of these things, you can see that the red's close to the blue here and lower than the blue elsewhere, and sometimes non-existent. And I'll have a little bit more to say about that in a few minutes. Just that these 2 extra 5' Gs somehow seem to add to specificity. Keith Jung's lab made shorter guide RNAs and showed that you make guide RNAs and instead of being 20 or 17, 18, or 19, they still work pretty well. 17 didn't work so well. They make the same kind of indels, and these shorter guide RNAs can stimulate homologous repair just as well as the full-length guide RNAs. But they're more specific. So back to this GFP assay, where we're looking for mutation knocking out GFP. This is with the full-length guides on top with single mutations or double mutations, and the truncated guides at the bottom. Had some effect on this very permissive guide RNA, but with this guide RNA that was already somewhat sensitive to mismatches, it pretty much eliminated activity with a single mismatch using the shorter guide RNA. And this again was sort of intermediate. I don't see any reason to do that. And going to the guide-seq, this is the standard guide, and this is one of the guide-seq. This is the one I went through for you before. Only 11% of the sequence reads came from the design target. With the truncated guide, now 30% of the reads come from the design target, and you've minimized the number of reads from other targets. So it does have a good effect on specificity. Both the Jung lab and the Zhang lab made modifications to the Cas9 protein that have improved specificity. So this is something from this paper from Ian Slaymaker. So when the guide RNA directs Cas9 to its target, the RNA binds to one strand, and then we have this display strand we talked about a little bit before. And although the crystal structures that had been published to that time didn't show the display strand, you could see that there was a groove that had kind of a lot of positive charges around it, which was a plausible place for that display strand to sit. So what they did was they began mutating some of these positive charges that had nominal contact with the display strand. And this is individual mutations, and you can see that the on-target efficiency for many of these was comparable to the wild type, just making single amino acid changes, but often the single changes reduced the cleavage at secondary targets. And then down here they began combining some of these single mutations where a positively charged residue is substituted with an alanine, and they came up with 2 candidates, one called Enhanced SP Cas9 1.0 and 1.1, that have these 2 mutations and one or other of these, which have very high on-target activity and very low off-target cleavage. And this is just showing using these single nucleotide substituted guide RNAs that the wild type is fairly permissive. Making a single mutation makes it less permissive, and these triply substituted ones make almost no cuts, no indels at this target, even with a single mismatch distributed through the guide RNA. In Keith Jung's lab, they made mutations in residues that actually contact the DNA-RNA hybrid and came up with similar results. So if you're reducing the affinity for the nucleic acids on the Cas9 protein, came up with quite similar sorts of results This CRISPR-Cas9 high fidelity 1 has 4 substitutions at these 4 positions that contact the DNA or the RNA strand and contact phosphate residues. And this has high on-target efficiency and low off-target efficiency. And they did guide-seq with these as well, and you can see that wild type compared to high fidelity, you're getting almost no reads at these secondary sites from a guide-seq experiment. And this just shows that they've made some other changes as well. So these 3 types of changes, the truncated guides, the unmatched 5' GG, and these mutations in the Cas9 protein may all work. It's plausible that they all work by reducing the overall affinity of Cas9 for the guide RNA target complex. And so there's excess affinity, which is why the secondary sites are getting cut. And reducing that reduces that cleavage at the secondary sites. And we call it excess because you don't need that much affinity to hit the design target. And the extent to which this is going to be effective is going to, and which of these things you might want to do and how much you might want to do, will certainly depend on the natural affinity of the complex at the target, which will depend on the guide sequence, may depend on other features of the target itself. So this may something that may have to be titrated for a particular target. Last thing I want to say, I don't want to cut into Luke's time, is that the CPF1 homologs, particularly the ones called AS and LB, Acetoaminococcus and Lactospiraceae, these seem to have inherently higher discrimination against mismatched targets than the CRISPR-Cas9 does. So there are data from Jinsoo Kim's lab that support this. This one also is showing that with both the AS and the LB-Cas9, you get additional specificity by using RNP delivery compared to plasmid delivery. And these are just sequence logos showing that at least a subset of targets, the LB-Cas CPF1 really cares about the nucleotide identity at every site throughout the RNA-DNA hybrid. AS CPF1 a little bit less so, and they are both better at discrimination than the CRISPR-Cas9. And this, a little more data from the Kim lab. Another thing that's interesting about this is that they found using each of these, the LB-CPF1, the AS-CPF1, and the CRISPR-Cas9, that when they used TIDE, you may remember TIDE, to characterize the indels that were made, that in 2 different experiments, each of these gave characteristic indels, just a characteristic family of indels. And you can do the experiment 4 times, 5 times, and you'll still see, like this one gives a +1 insertion, -1 deletion, and a -9 deletion in this particular target over and over again. So there's something about the target, maybe the microhomologies, they will certainly contribute to this, but maybe there's something else going on that governs what indels you get. And this is from Keith Jung's lab. Ben Kleinstiver was a very busy boy. He's making all those PAM variants, as well as looking at CPF1. There's a crystal structure of CPF1. And if you're looking to fix the specificity when you're targeting a particular gene, these are some guidelines. Design a new guide RNA. Maybe your guide RNA just has too many alternatives in the genome. It's going to go get some of them. You can try the paired nicking. This is quite effective. Not very many people have picked up on it, surprisingly to me. I think that's because the raw specificity is good enough for most people at this stage. 5' GG, I don't know how broadly that's been tested. The truncated guides give an improvement in specificity, except in cases where that truncation removes some of the mismatches that you're depending on. So if there are mismatches out at the 5' end of the guide, and you shorten the guide, you don't have those mismatches anymore. The Cas9 mutants seem to me very promising. CPF1 has this inherent specificity, and as I said just quickly in passing, this RNP delivery seems to help with specificity as well. So I think that's the end of that.